Okay, so this is going to be, oh, we have actually a few minutes, so I better wait before I say anything. I think we are recording. So yeah, so since we're recording, I did want to mention this next assignment, the peer review assignment, that's due between now and Sunday. And again, don't forget to have a look at this, but again, I'm asking you to select an article and then, okay, now I'm resuming recording. Thank you. Thanks for letting me know. Yes. So I'm wanting you to basically critically review the science, the experiments to see, if that would be the approach that you would take. If you think that they did a good job, especially did they include the appropriate controls? Did they provide enough background to make the audience, the reader, understand what the gap in knowledge was? Did they state a hypothesis? Did they have a good approach to this particular problem? And then, identify any strengths or weaknesses, things that you might consider that you think that, well, maybe they should have done this instead of that. And then determine whether or not you as a reviewer would accept that article. You can accept it, you can reject it, you can accept it with major changes, minor changes. Those are the kind of the typical, when these letters are sent out to the authors of these manuscripts, this is what they're receiving. And Dr. Mwangi and maybe others in the room have had this experience where you get this letter and it's either your publication has been accepted or rejected. Often that they provide you with feedback so that you can address what those weaknesses might be. There might be new experiments that might need to be performed or clarification sort of things. So I just want you to kind of be in the driver's seat as the peer reviewer, which is very different than actually writing a manuscript for peer review. So you're sort of in the driver's seat to sort of critically evaluate the article, whatever that article might be. And it does need to be centered on immunology, for goodness sakes. We are in that class. So Dr. Mwangi, you probably have some words of wisdom. Have you reviewed quite a few articles?
I'm actually editor-in-chief.
Oh, yeah, you've been an editor-in-chief. Yes. So I'm
currently an editor-in-chief of the veterinary vaccines for Elsevier. So yeah, send your papers. I'll assign them to my sub-editor. Yeah, so I review for a lot of journals. Yeah, over the years, I've reviewed for a lot of journals, and right now, yes, I'm editor-in-chief, and then I'm also a section editor in a few others.
Wow. Okay,
so,
you are a busy person.
Yes.
Lots of reading.
Yes.
So this is kind of helpful, especially if some of you are going to be submitting papers. This kind of helps you sort of understand what reviewers might be looking for, and how they sort of look at it from their perspective, give suggestions, that kind of thing. So it's just an exercise for you to kind of understand that side of things. We've talked about a lot of different papers and experiments in different papers. You presented on paper, so now it's your turn to kind of be the reviewer of some of these papers that are submitted. And as you know, some of the papers are good and some of the papers are not so good. And, so this kind of gives you that chance to kind of explore being a reviewer, because you will be asked perhaps one day to be a reviewer for an article. And so it's kind of good to get some of this experience as well. So anyway, it's just yet another good experience for you. Any questions on the peer review assignment? And also, again, don't forget to provide the citation for the journal article, or you can just submit the journal article along with your peer review assignment. Any questions before we get started? I know Dr. Mwangi has more to cover on mucosal immunity as well as cancer. So it's cancer immunology. So with that, I will go ahead and turn it over to you, Dr. Mwangi, because I know you have lots of materials. And also if there's any follow-up questions from the last two lectures from Dr. Mwangi, because we covered, hypersensitivity, and we covered mucosal immunity, which is really interesting. Are there any questions, follow-up questions for Dr. Mwangi?
So I've shared my slides. Can you see that?
Yes, we can.
Okay, let me put it in presentation mode. Okay, yes, thank you. So let's pick up from where we stopped, and we're talking about T-dependent IgA class switching in the gut. And the reason why I'm starting over here is to tell you that this is the slower process by which you get IgA. It takes a little bit longer if you now go via this route of T-dependent IgA or T-cell dependent IgA class switching. However, there is a much more robust, much more faster way of actually inducing antigen-specific IgAs via this route, which is T-cell independent IgA class switching in the gut. And comparatively, this mechanism really contributes much more, and you're going to see the reason why. So in this case, what happens is that antigens that derive from the lumen, they are detected by the cells, the lamina propria. But this is subset that are involved here are highly endowed with pattern recognition receptors. The example used here is TRR. These DCs are able to drive the following mechanism of B-cell isolate switching. And they do so by secreting multiple factors. One of those is called EPRIN, which stands for a proliferation-inducing ligand. Another very, very important molecule they secrete is BAF, or B-cell activating factor of the TNF family. You also have this subset, secreting TGF-beta. Taken together, what happens is that these cytokines and other factors that are not listed here, there's quite a number of them, okay? Only these three are shown here, there's quite a number of them. Once they have been stimulated by this route, the secreted factors do have an influence on resident IgM positive B cells that are actually quite many in this region. Where do they come from? When you guys talked about B cell autogeny, did you talk about B1Bs that initially come from the heparos, okay, at the fetal level? Now, what happens is that the stem cells that initially came from the liver, they migrate out from the liver, they're no longer there. They go into the gut. And they are self-renewing, and they end up now driving massive production of IgM positive B cells. One of these B cells, what they have is that they have generic BCR, which means they tend to behave more like innate immune responses, because these genetic receptors can be able now to have immediate recognition of certain common antigens, and then they're able now to respond. But there is also limited ability for these actually now to undergo class switching, not class switching, affinity maturation, VGJ comment, and so forth. The key point here is, under the influence of these factors secreted, the B cell is stimulated to switch from making IgM to IgA. And of course, from there, again, under the inflammation conditions that are transitional or transient, because now you have a pre-existing stimulant, in this case, a pathogen, you're able now to have generation of memory. You also have generation of plasma cells. And plasma cells that are now generated from here, again, they go through the cycle that I showed you, they home back into the gut. And then they become perpetual IgA secreting factories, and they're able now to participate in immune protection. So, to emphasize that point, this is real data. Looking at a section of the gut, and you can see now, if you stain for IgA and co-stain for IgG, you can see that the overwhelming number of cells here are secreting IgA, and mostly these are derived from the second pathway of the T-cell independent pathway that we've talked about. So the within the gut, we know clearly that it mediates or it confers protection within this critical part of our bodies. Is how do the IgAs get from the lamina propria to the lumen? So it's actually a very simple but very neat mechanism, whereby now the secretory IgA, which is dimeric, and what makes it a dimer, that it has J chain, that actually brings it, these two monomers together. That dimer is what is recognized by a very important chaperone or carrier protein or carrier molecule called poly-Ig receptor, which then binds now to the dimeric IgA. This poly-Ig receptor has these traits whereby it's able now to shepherd the dimeric IgA into and mediates its entry into the mucosal epithelial cell, and then while it's in the cytosol, direct it as a chaperone towards now the lumen. And then once it gets there, it anchors. You have the leader domain of this poly-Ig that then gets anchored on this face of the lumen, and then you have now cleavage or protein cleavage of now this lead domain, and that's how the dimeric IgA is channeled from the lamina propria into the lumen. And once it's there, it retains the rest of the poly-Ig receptor. The question is, what's the function of the poly-Ig receptor domain or molecule that is left bound to the J chain? Why is it important? Besides now mediating the transportation through this pathway, it is this chain fragment that actually protects the dimer from degradation by the proteases which are there. And that's why it's able now to hang around there much, much longer to be able to mediate its biological function. Okay. All right, so switching gears a little bit here and just giving you an overview of the intestinal villi, we talked a bit about components such as the intraepithelial lymphocytes, as depicted over here. We also talked about the presence of various lymphocyte subsets such as the CD4s providing help with the B cells. Then we also talked about the crypt epithelium, whereby it's reaching parent cells that end up secreting lysozyme and defenses. Now, what is not shown here is that this region is also rich in mast cells. And what happens, I have this this slide here is actually now give you an overview of events that happens here in the face of infection by intestinal worms. Because all those T cells, B cells, and all those, they play a role, but they play different parts of the role, and you're going to see how all this comes together. Now, the roundworm has adopted a mechanism of actually lodging itself into the epithelium so that it can have a mechanical anchor and then end up enjoying free food, free oxygen, and free everything. That lodging is strategic because, again, it's able now to be able now to acquire all these traits, all those its needs as it lives there. Now, the question is, within the gut, which are some of the important events that take place of an infection? Because the gut is not just going to be passive. It does something to try out to get rid of the worm. So one of the things that happens is that now you have antigens that are being shed from these roundworms. And these antigens, because of the signatures, the pumps they have on them, they tend to induce a B cell response that is associated with a Th2 immune response that promotes IL-4 and, of course, the generation of IgE. Now, that is one aspect of, or that's one outcome of this immune response cascade, such that now the IgE binds the mast cell. And then what happens is that the mast cell, in the face of or the presence of antigens now derived from the worm, can get triggered. And we talked about the granulation of mast cell that results into release of histamine. Histamine is very important here. And in fact, the gut is quite rich in a number of these mast cells. And this histamine ends up now perturbing that microenvironment to be able now to create some movement that results into these epithelium cells here having fluid leakage and so that, and of course then opening the gaps in between the epithelial cells in an attempt to get rid of this guy here who is taking a ride for free. Okay, so that histamine is what now drives the vascular permeability, smooth muscle construction. So you're looking at quite a number of events here. Perturbation, creating some smooth muscle to attempt to get rid of the roundworm. But on the other hand, the roundworm is not going to get it, that it's not going to get kicked out that easily. It's home. Okay, so of course it has a mechanism by which it allows itself to stick around and be able now to counter the effects of this. So what else then happens? Even though now you're going to get free accumulation over here, what happens is that you're going to have other mechanism coming into play so that now you can have a successful expansion. So you're going to have synergistic reaction or synergistic immune responses coming together. And one of those, some of the additional immune components that kick in is upregulation of IL-5 and IL-15 and IL-13. Remember when we talked about type 1 hypersensitivity, I told you that IL-5 is very important. It is critical because it ends up now coming into play here because it not only now recruits, but it also is involved in activation of eosinophils. Mast cells by themselves, they can do whatever they want over here, but they might not really effectively get rid of this worm. But now when you have eosinophils, the game starts changing a little bit. Okay, so we're going to see the mechanism by which these eosinophils really do mediate the killing of the roundworms in the next slide. But you also might end up having a role for complement that is triggered by antibodies generated against surface determinants of the worm. Complement by itself might not actually poke holes on the surface of the worm because they have a tough surface. But the byproduct, remember like the C5A we talked about? It is what? It is one of the molecules that can also activate the mast cell. Do you see now the synergy of how these components are coming together? All right, so the strongest powerful tool that this immune system have here with regard now to taking care of the roundworms is ADCC, antibody-dependent cellular cytotoxicity of parasites by eosinophils. Meaning that now you have the Th2 T cells that we talked about that are not only now promoting the B cells to secrete the IgEs, but they are also secreting IL-5 that recruits and activates eosinophils, such that in the face now of the antigen-specific IgE or roundworm-specific IgE, you have now ligation of the receptor of the Ig receptors on the eosinophils, and that ends up now resulting into in eosinophil activation. And the molecules or the factors secreted by eosinophil are the ones actually which does the job. Which ones are they? It's an array of them. These eosinophil degranulation release components, they are quite powerful. Major basic protein, eosinophil cationic protein, phospholipase D, phospholipase, all those and many others that are not shown over here, they are very toxic to these helminths. And what happens here, as shown here, is that you can see these helminths over here. All these surrounding these helminths, these are eosinophils. So they end up now mopping it and end up now degraduate these factors. And it's these factors that end up now in the holes on the helminth or the roundworm and mediate its death or promote the killing of the killing of the roundworm. So I wanted to pause here and find out before I move to the next stage whether there is any question here. Now, the role of ADCC that is mainly now driven in this side by eosinophil is also, it also receives some help by recruitment and involvement of some neutrophils and macrophages. But the major player here is the eosinophils. Okay. And in fact, if you had to rank them, then of course, remember the way we talked about starting with the mast cells, the eosinophils come into the scene. Now the inflammation around there again enables or even promotes the recruitment of eosinophils and the macrophages. But again, those are secondary compared now to the role played by the eosinophils with regard to ADCC. Okay. All right. So talking about the mucosal surface, it's not just about now, we've just looked at one example of how it deals with a pathogen such as the roundworm. Okay. And we talked about how antigen-specific IgAs are produced, and those could be through invasion of bacteria or viruses or viral pathogens within the gut. But the gut also has another major function. It is rich in Tregs, and these Tregs, they have a major role to play. Otherwise, if they don't, you have a problem. Folks who have Crohn's disease, because of the perturbation or imbalance or hyperactive gut. And what happens? The presence of Tregs that end up mediating secretion of significant amounts of immunosuppressive or anti-inflammatory molecules such as interleukin-10 and TGF-beta and so forth are important because they now modulate Th2 immune responses, Th1 immune responses, and Th17 immune responses, especially after these effector subsets have actually done their job, so that then they can be able now to restore the balance or be able now to bring everything back to normal. So if that mechanism does not or fails to kick in, then you have this gut condition that is always on a state of inflammation. And folks who have unstable guts, they have very uncomfortable life. So the role of Tregs here cannot be overemphasized. It's quite important. So with that, I think that wraps now the brief overview of the mucosal immune system. And here you have a list of your learning outcomes, which are actually brief, but then you can be able to expand this by again reviewing the materials that you've covered and be able now to expand this for your own reading and studying, and also serve as a summary of what we've covered. So any questions on the mucosal immune system?
Right, any questions? I was just going to ask you for the let's see, the independent T cell independent antibody responses, the IgM. Is that did you say the B cell receptor of IgM is how is that? Is it specific to the antigen, or is it sort of a, when does it become formed to be specific for the antigen if it's not really, if it's sort of bypassing T cell influence?
Okay, so to drive let's say recombination and affinity maturation and all that, at that point, for these T cell independent B cells, you have a significant population of those cells generating what are called natural antibodies. They have this generic IgM or BCR that have capability of actually recognizing antigens weakly, but in a very generic way.
Gotcha.
Why are they important? They're important because they form a first line of protection. But because of those factors secreted by the DC that we talked about, it's able now to drive not only isotope switching, but also to some degree, some level of recombination and affinity maturation, affinity maturation, to be able now to make what you can call higher quality B cell responses. Which, of course, then when they switch from IgM to IgG, over time, depending on the level of stimulation, they become better as they get exposed to the stimulant or to the antigen. So it's not that they are not specific. No, they are weak. A majority of the B1B. Remember when you studied your B cell autogeny? Yeah, a lot of natural antibodies are derived now from the B1B subset, and the stem cells now that actually as a person grows, they relocate now into the gut and be able now to repopulate the B1B, the IgM positive B cells within here. So, but again, they are not as robust as the T cell dependent or B cells that are derived through with the help of T cells. But these guys, they compensate that one by sheer numbers. There's a lot of them. And then gradually, as they get restimulated, as they keep on getting sensitized by the antigen, they end up now sharpening or be able to improve their ability to recognize their targets.
Yeah, that's so interesting. The mucosal immunity is like a whole other, we learn about the lymph nodes and all those, but then when you switch to the mucosal immunity, it's pretty impressive. I think that's more impressive than the routine one.
Yes, and remember now the pious patch is referred to both as what? A primary and a secondary lymphoid organs. The reason why it's primary is because remember the stem cells I talked about? They're self-renewing and they're able now to generate B cells. So that's why pious patches are referred to as primary lymphoid organ. And then all these are events that we talked about. Again, they do happen there in a very similar way that you would see, let's say, in a secondary lymphoid organ, like a lymph node or within a spleen.
Great, thank you. Any other questions? All right, off to cancer immunology.
Yes, immunobiology of tumors and immunotherapy. In previous lectures is that this is a very, very brief overview. And unfortunately, it doesn't even capture some of the latest technologies, some of the latest discoveries that we have. But you're going to see in general and learn a little bit about this subject by looking at these following key points that are listed here. And instead of going through them one by one, I think the best thing is to actually to dive in and actually now be able now to study them one by one. So, tumor antigens, how do they come about? So over the years, over the decades, it was actually discovered that tumor antigens are derived from different sources, one of those being mutated proteins, which is typically altered normal proteins that end up now getting out of control. That's one. Then it was also discovered that, viral oncogenes or viral-derived or viral products can actually drive tumorigenesis. Okay, that's another way that tumor antigens are generated. The third one here is a category of tumor antigens that arise because of what is termed as differentiation antigens. Okay, so proteins are dealing with specific stages of cell differentiation. Okay, and the fourth one here is when you have excessive amounts of normal proteins or production of normal cell proteins. The best example we can give here is the HER2 or breast cancer antigen, Okay, normal there's basal expression of that protein, but when it becomes excessively, you have an overproduction of that, then it falls within this category of too much. And then you have a class here of quite a number of proteins that are of unknown functions. In fact, cancer is the highest, if not, I think the most highest funded research on a global basis. A lot of resources have been pouring and no knowledge every day is coming out. But again, we keep on learning more information every day. And so what we're going through here is actually just brief. So, but let's step back and see some of the conditions that might promote formation of tumors. So this scenario here is explained by the fact that the mechanism by which we regenerate new cells in our bodies is not perfect. There are always errors being made. And even a very healthy person, you have some variant cells that arise. But because your body or your, if the person is healthy, the immune system is able to take care of those aberrant cells even without causing any problem. And you wouldn't know that has happened. So cancer tends now to exploit that if conditions are right. So there's this phenomenon called bystander effect. There's a aberrant cell that has this mutation or these changes, arises. A healthy immune system can get rid of it. However, if you are under stress, or there is inflammation that is coming from somewhere else. Inflammation could be because of infection, it could be inflammation because of other factors. That ends up now promoting the following events, whereby now the inflammation ends up now driving these transcription factors. The transcription factors upregulate chemokines, cytokines. And what happens is that the inflammation that arise end up now recruiting inflammatory cells that end up now promoting that one cell that could have otherwise been gotten rid of very easily. All of a sudden, it's sort of like a lifeline has actually been thrown away. It only needs to start dividing very slowly, 1 into 2, 4, 8, and all of a sudden becomes a problem. So inflammation is actually not good because inflammation has now been proven to contribute to formation of cancer. In a very simplistic way, that is what I wanted to communicate to you. Now, how do we know this? The best example here, if you look at certain adjuvants, and this is an aluminum-based adjuvant, they end up now causing this post-vaccinosarcoma in a cat. And simply what's happening here is that you have an immunogen formulated in an adjuvant, and what happens is that promoting excessive inflammation, and all of a sudden you end up having an outcome like this. All right. Okay, so you can end up categorizing malignancies in 2 ways. The first one here is what you can call you have malignant transformation that end up now resulting into either homogeneous or heterogeneous malignancies. How do those arise? So typically what happens is that oncogenic viruses end up driving formation of homogeneous malignancies. Why is that the case? Because oncogenes have got a propensity to get inserted in certain specific sites within the DNA, or they end up preferring to end up being involved in certain biochemical pathways. So they're very specific, they're very biased. And because of that, if they get, for example, if you have an oncogene being inserted into a certain site, then the resulting malignancy is actually going to be uniform. So that type of malignancy is actually quite easy to deal with because it is homogeneous. On the flip side, if you now get exposed to chemical carcinogens, radiation, and so forth, those carcinogens end up causing random events or random damages to the cell, such that you end up now resulting with heterogeneous malignancies. They're not uniform. Okay, and those are actually much harder to deal with because they have dissimilar antigens, and there's no single or a few antigens that you would say are going to be key markers or key targets that you can focus on. Okay, so this table here is a bit busy. It has a lot of information regarding examples of tumor antigens, but again, it sort of summarizes some of the information I've given here, and as listed here, and given as examples of type of antigen, then we talked about products of oncogenes, and others could be you having suppressor components. All of a sudden, they fail to function, which means they fail now to be able to mediate their function. And quite a number of examples are shown here. And I just wanted to look at a few here, like products of genes that are silent in most living tissues. Like what happens with the cancer testes and cancer and testicular antigens, and that are expressed in melanoma and carcinoma cells that do arise in these tissues. So I also wanted just to sort of focus on a few more important examples here that you can actually relate to, instead of now dwelling on a lot of those, on a majority of these. So these are the products of oncogenic viruses, and I think these are more familiar when you consider the example of the E6 and E7 proteins that are the key drivers of the cervical carcinomas. And we know, of course, right now, I think we have some vaccines that are actually working very well. But again, these products of oncogenic viruses, they have been well characterized, and they are known on how, or the mechanism by which they deploy people now to induce tumorigenesis. The other one is Epstein-Barr virus, overlapping protein, EBV, that end up causing EBV-associated lymphomas, and so pharyngeal carcinoma. And again, right now, we don't have a good vaccine for EBV, but there's quite a number of candidates in the pipeline. Now, one of the well-characterized oncogenic protein is the so-called SV40, which stands for simian virus, large T antigen. This SV40 large T antigen is very important at inducing tumorigenesis. And what has been shown that if you just transfect this gene encoding this into a cell, and a primary cell, then you can actually immortalize it. That's how we immortalize cells, primary cells, by just transfecting with this. So, I've just sort of glossed over a few of these just to let you appreciate the diversity and different types of tumor antigens. But I wanted to switch gears here and talk about some of the management options that are available. So if it's late in the process and you have formation of solid tumors, the normal way of dealing with this, or the most common way of doing it, has been actually have surgical removal of the solid tumor. But of course, by the time you're having a solid tumor, you have already had metastasis. The cells have actually spread within the body. So you have to follow that now with treatment, chemotherapy, radiation, to be able now to kill those residual cells. Now, more contemporary strategies are actually now using more focused and more targeted approaches. And some of those approaches are now utilizing well-characterized, potent antibodies that are actually specific and target these tumor antigens. And which means the antibodies can go into the patient and hunt for these cells, because once those antibodies bind, then they're able now to mediate killing through complement activation, ADCC. They can also actually end up mediated killing via the FC-mediated phagocyte activation. The other immunotherapeutic approach is utilization of CTLs, and I'll tell you a little bit about this. And the other one here is inhibition of Tregs. And that strategy arose when it was realized that cancer cells, and you're going to see some examples here or some information here, that they end up thriving by actually shutting off immune responses, specifically T cell responses, to be able to for them to thrive. The ongoing and actively under research area is generation of now better immunomodulatory mechanism. Like, for example, using an immunostimulant to be able now to switch from immunosuppressive microenvironment to pro-inflammatory microenvironment that will promote the killing of the tumor cells. But then there's also again development of cancer vaccines. And you're going to see briefly here some of the examples of how folks are going around doing this. So to get there, let's talk a little bit about the mechanism by which tumors really fool around with the immune system. Now, typically, what happens is that if you have recognition of a tumor cell by T cell, these end up now resulting into T cell activation, and then you have subsequent killing of the tumor cells. However, what happens is that one of the easiest mechanisms by which tumor cells do evade is that you have this cell, cancer cell, that does not express a cancer-related marker, or you have absence of display of cancer-specific epitopes on the surface, which means that T cells cannot recognize it. That becomes very difficult. The same thing would be said with tumor cells, or of a cancer cell that is not expressing an antigen surface that could be recognized by an antibody. So if that antigen is hidden, then antibody cannot see it. So antibody-mediated killing mechanisms are rendered useless. So that is one mechanism, failure to produce tumor antigen. The second one here is that cancer cells have figured out the mechanism of actually, or you can end up having a tumor cell that arises because of changes that occur within MHC molecules, which means that you could have this cancer cell producing as much tumor-specific antigens, but since it is not expressing MHC molecules, then there is no display of the antigen to the cell surface for recognition by the T cells. Last but not least is that tumor cells are notorious. They have really figured this out of producing or overproduction of immunosuppressive proteins such as TGF-beta, and these programmed cell death receptor ligands such that they end up now inhibiting T cell activation. So, in a nutshell, all the information I've told you here is actually summarized here, whereby evasion of the immune system by tumor cells, whereby you have loss of expression of antigens, okay? And you could also have mutations within the MHC molecules or the machinery that actually loads what? The peptides onto the MHC, the TAP, okay? So that can also end up leading into that scenario. The other one is secretion of immunosuppressive cytokines that I've talked about. And you could also have, and this one I did not mention in the previous slide, is that you could have a tumor cell that is producing a lot of tumor-specific antigens, but these antigens are expressed in such a way that they are in a heterogenic form, okay? Which means that you end up now with a depressed or lack of costimulatory molecules. So which means they're actually dampening or depressive in the sense that they're making the immune system become non-responsive. Okay, so tumor cells can also promote or do promote generation of Tregs, and they end up doing that by producing this program death receptor or PDLs that bind to the receptor of T cells. So that is another mechanism. And last but not least is this mechanism called antigen masking, meaning that you have an antigen on the cell surface. B cells can make antibodies against, but what happens is that they end up getting this generation of antibodies that are biologically non-functional with regard out to ADCC, complement-mediated killing. So, which means that the antigen end up now being masked. And since you're having B cells producing those antibodies, the tumor cells, every time it divides and you have this antigen displayed on the cell surface, that antigen gets masked. And so that's how those cells end up evading destruction by the complement system. So, looking a little bit in brief, some of the key cellular components that play a role in tumor destruction. We start off by looking at the role of phagocytes, but not just phagocytes, macrophages, but not just macrophages, activated macrophages. Activated macrophages, they have, of course, they end up now being responsive to the presence of tumor antigens. And how do they end up now catalyzing the events that happens? One of the mechanisms they do that is by secreting interleukin 12 and TNF-alpha. Those two molecules are actually very important. Interleukin 12 is a fuel that actually drives NK cell activity, stimulates NK cell, and the NK cell that is stimulated, in return, it secretes interferon gamma. So there's that synergy. The interplay between those two is important. Now, the interferon gamma empowers the macrophage to become highly activated and endows it to be able now to not only bind to tumor antigens, but also take them up and destroy them. On the other hand, the activated NK cell by itself can recognize tumor antigen and end up killing the tumor antigens directly, or tumor cells, not tumor antigen, cancer cells directly. And the mechanism that they utilize is fast ligand. Okay, you also have now a generation of this, because they're lytic, they also end up secreting afferent and grazine that end up now killing the target cells. Now, that early stage, or those early events that I've talked about, activation of macrophages, activation of the NK cells, can create a microenvironment that is conducive for the phagocytes. Particularly in this case now, you have to have a role of the T cells coming on board, picking up some of the debris, some of the tumor antigens, and presenting those now to T cells, so that now you have priming of tumor antigen-specific T cells. And once they show up, they end up really being a game changer, because these T cells can be stimulated to differentiate to become CTLs. You also have potential of now generating CD4 T cells that are actually secreting much higher levels of interferon gamma. Okay, and so they end up now modifying the microenvironment such that they can end up now creating a microenvironment that is conducive for destruction of the tumor cells. But again, remember what I told you. The tumor antigens as matter, in the sense that they want to shut off these events so that they can thrive, right? So, historically, there has been a lot of efforts that have been put in place and pursued to mitigate or manage cancer. And so we just wanted to look at some examples of anti-tumor immune response or immune strategies that have been pursued in the past. And one of those that were pursued earlier on for folks to figure out, okay, tumor cells, some of them, are very prolific in dividing. If you can figure out of looking at an immunotoxin, and this could be bacteria toxin or it could be other toxins that derive, some of these could even be chemical toxins, and then you target those toxins into tumor cells, you can end up now killing the tumor cell. But again, use of immunotoxin end up also having collateral damage because you can also have bystander effect, whereby now you have healthy cells also being damaged. So other folks, what they've done in the past is now using immunostimulants, and some of the molecules that were utilized were cytokines. Okay, when it was discovered, oh, interferon gamma can actually activate macrophages very well. Certain type 1 interferon, type 1 interferons can also be able to mediate immune response. So they end up now trying to pursue those, okay? Interleukin 2 and so forth. But it turned out to be use of cytokines was not ideal because it required high levels of the cytokines. And with those levels, they end up becoming toxic, okay? So other folks, what they've done is looked at ligands that stimulate NK cells so that now you can have NK-mediated killing of the tumor cells, or using stimulants that are targeted to macrophages. For example, ligands that macrophages have receptors for to be able to activate them. And once they get activated, then their function or they're endowed with an ability to be able to destroy tumor cells. The last one here is use of stimulants, immune stimulants, to be able now to switch or to be able to skew the immune response from all the microenvironment from a depressed microenvironment into or from an anti-inflammatory microenvironment into a pro-inflammatory microenvironment to be able to promote generation or to be able to promote activation of both phagocytes and other players to be able to clear the tumor cell. So with that, I just want to pause for 1 second and find out any questions so far, because I want to talk about the earliest experiment that was done to really demonstrate that and demonstrate the role of immune response, specifically T cells, with regard now to tumor clearance. Any questions?
Any questions?
So in this very early, the earliest, this was, I think, in the 60s or 50s, what they did is that they were able to induce a tumor using a chemical carcinogen. And what happened is that they could now resect or remove this tumor. And what they did is that they took this tumor and ended up now transferred these tumor cells into an, so what happened is that they induce the tumor, but then they come and mechanically, just surgically remove it. And they did, this model here was such that they were able now to remove the tumor, and then they ended up now getting majority of it so that there was no residue. So what they realized that the mouse was able now to heal, but then when they took that tumor tissue that they had removed and then transplanted it back into that mouse, there was no tumor growth. So from the time that they induced it to the time they removed it, something happened, such that now that mouse was now in a position to clear those same tumor cells when they got reintroduced. To prove it further, they took those tumor cells and they ended up injecting it into a naive mouse. And they saw, hey, wait a minute, there's nothing wrong with this, the cells we are getting now from this tumor, because if you transplant them into a syngeneic, a mouse from the same MHC background, you're able now to induce a tumor, and that tumor end up now thriving. Okay, and if you don't do anything else, yes, it might end up now killing that mouse. So the question was, what was happening when now you expose this mouse here to a carcinogen, and then you end up now removing that tumor? What is going on here? Long story short, they actually demonstrated that if you now induce this tumor and you look closely to this animal here, you can actually find some tumor-specific CD8 T cells. That if you now, they took those here and expanded them, and then adaptively transfer that to a naive mouse. And then what happens is that then they challenge that mouse with the tumor cells, and that mouse was able to clear those cells very, very well. This was the first demonstration that indeed tumor immunity can be induced, and specifically CD8 CTL mediated destruction of the tumor cells. Okay, so with that in mind, folks now started thinking of how can we be able now to induce immune responses against tumors by using that knowledge. So it was understood that one of the possible mechanisms that happens is that given the tumor cells by themselves, they are very poor antigen presenting cells. And given the fact that you require a much better APC, such as an adenosine to do that, prime antigen-specific T cells, the envisaged route or mechanism by which this happens is that adenosine cell has to take up this tumor cell and be able to process it and present epitopes, both to CD4s and to CD8. To CD4s to be able now to prime tumor-specific CD4s that can then provide help to the DNA cells in terms of contact-to-contact co-stimulation, or the CD4 secreting cytokines and chemokines and other factors that end up now promoting and enhancing the ability of adenosine cells to prime and expand tumor-specific CD8s. The reasoning behind here is because induction of CD4 T cells is much more easy. That is easily done by adenosine cells, but priming and expansion, especially priming of the CD8, the signal threshold that is required is much, much higher. But once now they are primed, and in the presence of peptides presented by image class 1, the CD8 can actually now expand, recognize the tumor antigens, and be able now to mediate the killing. With that in mind, folks now decided, okay, how about if we take the cells, we transfect them with a tumor-expressing antigen, okay? Or if we take the cells and pose them with tumor antigens, they were able now to achieve some level of success, because depending on the conditions that you do this, there are certain factors that you have to provide to be able now to create a microenvironment that is ideal for the DNA cells to be able now to prime CD8 CTLs and be able now to produce these effectors that can be able now to recognize the tumor antigen and be able now to lyse or kill the cell that is presenting or is expressing that antigen. The other strategy that was derived from there was that, how about if we take now the cancer cell itself, because we know that it's a poor at antigen presentation, there is lack of co-stimulatory molecule and so forth. So what they do is that they transfect the tumor cell with the co-stimulatory molecule, the B7, and they also include a plasmid that is expressing interleukin-2, okay? So what they showed here is that to some degree, that transformed or transfected cell can actually be able now to present some tumor-derived peptides. And the presence now of the B7 and secretion of IL-2, they had some success in driving generation of tumor-specific CD8 T cell responses. Again, this data here is presented in a very simplistic way. These experiments are not easy. They're not easy, especially now when you're conducting them in vitro. They're not easy. So, based on that mouse study that I told you, now more contemporary approaches are being done in quite a number of hospitals, like Baylor College of Medicine and so forth, is that there's what you can call now a tailor-made vaccine mitigation strategy, whereby if a patient is detected or a tumor is detected in a patient early enough, what they can do is they can go and resect that tumor. And by then, depending on the level of they're able to detect that the host has generated some level of T-cell responses. So they get a tumor, and they also get some lymphocytes from this individual. Then they stimulate the lymphocytes or the T-cells in vitro. Okay, there is a combination of factors, growth factors and so forth that are involved here, whereby they end up now promoting expansion of those T-cells. And once they take off, they do thrive very well. Now they have to expand them to reach certain numbers, and then they're able now to transfer them back to the patient. And those T cells not only now are going to have that effector function, they're able also now to confer long-term memory in that host. And they've had some success in actually clearance and protection against the recurrence of the cancer. Okay, so that's an example from the human field. We wanted to look at this example from canine melanoma vaccine, but I just wanted to warn you that this is not a very good example. Initially, it was thought that it was one of, but it turned out to be it's not that very effective. So how did they go about doing this? So what happens here is that this tends to work very well in dogs which have already undergone stage 2 to 3 of melanoma, for which they've been able now to control the disease. And how do they do that? They can go and surgically remove the tumor. And then they end up now testing the draining lymph nodes to make sure that they're negative. Because what happens is once you start having metastasis, those cells end up now starting, and you can actually be able to track them into these lymph nodes. So by now following that, then you can, if you capture them early enough, you are going to find them in the some or a few of the draining lymph nodes. And then what you have to do is that you also have to remove those. Now, those dogs which have gone through that process and then they are being treated for irradiation, this cancer canine melanoma vaccine has been shown to have some benefits to some degree. So what's the target for this? The melanoma is characterized by expression of this tyrosinase protein, which is done. I mean, it occurs in larger amounts. But what happens here is that they could not use the canine tyrosinase because it's so it's not immunogenic. The dog is already tolerized that. So what they did is that the initial assay, they say, okay, let's use canine tyrosinase antigen and see whether it's going to work. Of course, it did not work because again of the reason I've told you. But then when they use the human tyrosinase antigen and deliver these in the form of a DNA vaccine, they're able to show some benefit. But overall, when you look at the total number of dogs which have gone through this process, the benefit is not, or let me put it this way, the extension of life or the benefit derived from this treatment or this approach is not that significant. So the initial excitement, over time, it has waned. But it showed that this so-called xenogenic DNA immunization strategy could work. Okay. All right. So besides now all those strategies that we talked about, there are other mechanisms that have actually been shown to work and work very efficiently. BCG, Bacillus calmette-guerin, which is what? What is BCG?
Vaccine for tuberculosis.
Exactly. In the United States, it is not approved as a vaccine for TB, but it's approved as a treatment for cancer because it works very well. And how does it work? BCG is rich in pattern recognition, pathogen-associated molecular patterns. So it activates phagocytes very well. Application of TNF alpha, interleukin 2, nitric oxide, and so forth. This BCG-based treatment is actually called Therasis, okay? And it's very effective at mitigation or management of human melanomas, bladder tumors. In fact, quite a number of folks who have had bladder cancer, it has worked, okay? So the other treatment option here, or the other management option, which exploits the innate immune responses, is this product called regressin. Regressin is nothing else but cell wall extract from a bacteria that ends up actually now working by stimulation or activation of the immune system, to the extent that it's able now to result into clearance of these equine sarcoids. And you can see that in this horse, before and after, you can actually tell it's a major difference. So again, it's still utilizing the innate immune mechanism. Another one here is use of mechanical tissue damage to be able now to induce inflammation. So exactly what's happening here. So this horse here has this bovine papillomavirus warts. And then if you use a hemostat, and by the way, this is still in practice, and it works. So you squash these warts with the hemostat, and you end up now causing release of danger-associated molecular patterns that end up now inducing inflammation. And again, the phagocytes and all that, those events takes place, and they're able now to mediate the clearance of these warts. Again, you talk to any horse owner, and if somebody who has dealt with this, you're going to say, yeah, it works. It does work. Okay. All right. So more recent, and for which about what, 2 to 3 years ago, some folks got they got what? Nobel Prize for this. Okay. They end up discovering, hey, wait a minute. What happens is that tumor cells express programmed cell death, programmed death receptor, the PDL1, which is a ligand, that when it binds now to its cognate receptor or T cell, which is the PD, or programmed death receptor, the interaction here is quite powerful at immunosuppressing the T cell. So they discovered, hey, wait a minute. You remember those incidents I told you about, that mouse model, and then the one for the human where they're able to reset? What happened is that for the humans, what happens is that you end up having low levels of anti-specific T cells, but they exist in low numbers and they don't thrive. They're being immunosuppressed. These folks, what they discovered is that if you use, let's say, a monoclonal to block either the receptor, as shown over here, or the ligand, which means now you're separating the T cells and this cancer cell. The T cell now is released from that suppression, and it's able to thrive, proliferate, be able now to increase in numbers, and then it kicks in and starts now killing the target cell. This is one of the strategies that is currently being deployed in the clinics, and it's working. Okay. Now, last but not least, here is that there's a list of biologics, tumor-specific antibodies that are actually working very well. So this list here is quite old, but it actually takes us back to those initial demonstrations that antibody or biologics based on antibodies can and do work very well. So one of the initial products that has actually been shown to work very well is this antibody against human CD20. There's a product called rituximab, which is a humanized mouse monoclonal antibody against beta lymphoma. It worked so well, such that today, human B-cell lymphoma it's no longer a threat to the human population. It's a treatable, the first treatable cancer is this one here because of a monoclonal antibody. Again, if you now go and look at the breast cancer in humans, quite a number of monoclonal antibodies are right now in the market and they're working very well. So again, you see a list of monoclonals that are listed here. Bottom line is, out of about 10 leading products in the market, when you consider biologics and vaccines, about 7 to 8 of those that are actually top earners currently in the United States, the monoclonal based drugs. They're working very, very, very well. So you hear a lot of vaccines cannot match the returns and the impact of the biologics that are based on antibodies. And that field is actually exploding. Why? Because you can actually engineer antibodies, you can actually be able to tailor them to do certain things. And so it's very versatile. So what you need to do is identify that novel cancer marker, generate these nice antibodies against it, and then you're able now to trigger ADCC, complement killing, macrophage, and once you do that, it's being shown that it's working very, very well. Okay, so who wants to be rich?
Everyone.
Then come up with a novel cancer target, make some nice antibodies, and boom, overnight you're going to be in very good shape. Okay, so how about regard to diagnostic tests that are deployed? And again, this is just only brief. So what happens is that immunophenotyping by use of flow cytometry, immunohistochemistry, and so forth. But that requires now you have to have good what? Antibodies, good markers to be able to do that. But quite a number of these cancer cells end up following the category like the triple negative, that they are not having these unique markers yet. So what for diagnostic, you have to have somebody who is trained person who can then have this cytologic evaluation and say, I think we're having cancer cells over here. But then the other one is use of PCR. But the PCR really is deployed when you're already suspecting that, yes, the tumor cells is due to a mutation somewhere. Okay, and so then you have to have primers really that maybe span that place so that then when you PCR, you can be able to find that mutation. Because again, on a fishing expedition. Because if you don't know where the mutation is, you'll be on a fishing expedition, right? Okay, so with that, I have a list of your learning outcomes, and again, as I said before, again, this is only brief. So with that said, I'll entertain your questions.
Are there any questions? We've got a little time to ask questions on any of the materials on autoimmunity, hypersensitivity, mucosal immunity, cancer, immunology. Any questions? Hannah? I'm not sure if you are unmuted for a question. Do you know if there have been any monoclonal antibodies targeting animal tumors?
I think
Or has
yes.
anyone heard of any tumor monoclonals? Because veterinary monoclonals, as those of us know, are getting very popular these days in the veterinary side of things. So it makes me wonder, are we going to see more of this in the future for even tumors?
Right, actually we are working on one. We are working on canine CD20 because rituximab or the human anti-CD20 does not work in dogs. But we started working on it about 4 or 5 years ago, but we've been slow on that because of certain things. However, a company in UK which received billions of dollars, they just beat us to it. I think they have a candidate that looks promising, but we also have some candidates that we are evaluating here. And we are going to continue doing that. You require variety, because whatever they are coming up with might have certain level of potency. We don't know what ours will look like. And ours, we're producing these in various platforms. One of the unique platforms we have here, by the way, is use of cow antibodies. And the reason we do that is because cows make some unique, very, very strange antibodies that no other species in the world does. They're able to see certain antigens that no other species can do that. So we also have some materials that we immunize from rabbits, and those actually they recognize it. I'm saying this because we have data to show that we have antibodies that recognize in canine T-cell lymphomas very well. But then we have now to screen and be able to identify monoclonal targets or candidates that have potency or that are good enough to actually mediate the killing of these cells. Yes, so to answer your question, yes, that area has actually exploded. This company in the UK, the reason why they were able now to make such dramatic progress is because they were able to generate a dog in a mouse, canonized mouse. A mouse model that, like the way you have a humanized mouse, they have now a canonized mouse. So they were able to humanize that mouse and be able to produce 100% canine IG molecules.
Huh, wow.
Ours are going to be chimeric because you are going to be grafting them onto the canine backbone. So they have the machinery, they have all the tools to be able to move forward. And of course, the major companies in the US that are dealing with the companion animals, they have invested in that company. Name it, any company, Zoetis, they've invested in that company.
Zoetis, yes.
Yeah.
Yes.
So-
Yeah. So you might be rich really soon here, Dr. Mwangi.
Hopefully, no, of course. Well, if things had worked out quite as we thought maybe a few years ago, and again, it's simply because when we started doing this, attracting funding at that point was not, we were not even attracting major funding because, again, at that point, who really, very few people really cared about it. You tell somebody, say, yeah, when you come up, you come and see me, so forth and so forth. But again, we have some data here that's looking very interesting. It's a matter of now screening and see whether we can be able to identify targets that are going to be functional.
Yeah, that's great. That's great.
Dr.
Eric, did you have a question?
Yes. So when you say tumor, I think of earlier we talked about in a class the Tasmania devil about the facial tumor they have. So I just want to know if there are any monoclonal antibodies for the Tasmania devils for the facial tumor.
The Tasmanian devil. Do you know anything about the Tasmanian devil tumor?
No, I don't know anybody. And you understand how those tumors arise? You do, right? But I don't know. I'm not aware of anybody who has any therapeutic antibody against those.
So
But
Eric,
yes, that would actually be nice because really those devils have been threatened. They are at risk.
That will make you rich again.
Yeah.
there you go, Eric. So you can go ahead and start generating those monoclonals so we can cure these, or at least treat these animals that are affected by that.
Oh, and there might be a market. Those folks down there are going to give you some money because they want to reserve. Folks go all the way there to see those devils,
Yeah.
Yeah. Tourism, yes, the folks who want them preserved, so yeah.
Tourist trade, I guess.
Yeah. Transmissible cancer,
Transmissible
How to think of
cancer.
it. Yeah.
Bad.
Yeah.
That's a good thought, Eric. I like that idea. It's a great idea. Any other questions or comments? Follow up, anything like that? Okay, how about in the room? Any questions there?
Not at the moment.
Not so much. Okay, well, I guess that kind of concludes Dr. Mwangi's lecture series. We appreciate you lecturing very much. That was a really informative, a lot of great information that kind of brings everything together for the course. So so it makes it even more, a higher level as we progress through the course. We started pretty basic, and we've
Okay,
progressed to the level of this complexity of mucosal immunity, hypersensitivity, tumor immunology. So it really fits in well with everything that we've covered this semester. So thank you very much, Dr. Mwangi. We appreciate it.
welcome. I enjoyed it.
Yes, I appreciate it. And if any of you have any further questions, even after this class, you can send your questions to me and I can send them to Dr. Mwangi, or you can send them directly to him,
Yes.
and he'll be able to answer those questions as well.
Yep.
Because you might think of something after we sign off tonight. Okay, so I guess that is all for tonight. Again, don't forget about your peer review assignment. And then next week, we'll have student presentations on Monday and Wednesday. And then, of course, we've got finishing up quizzes and exams next week as well. So it's going to be a busy couple of days next week. So get some rest, and thank you very much. Hope everyone has a great evening.
Okay, bye-bye.
Thank you. Bye-bye. Have a good evening.